In this video, I'll be solving paper 4 variant 2, October-November 2021, Cambridge IGCSE Biology. 1. Figure 1.1 shows several villi from the ileum, which is part of the small intestine. A state the name of one other part of the small intestine. So ileum is the first part and duodenum is the second part. Figure 1.2 shows the tips of a villus in more detail. So here you've got the goblet cells, the microvilli, and the epithelial cells. B. The epithelial cells of the villi absorb nutrients by diffusion and active transport. 1. Describe how active transport differs from diffusion. So active transport is the movement of substances against their concentration gradient, whereas diffusion is the movement of substances down their concentration gradient, so from a region of higher to lower concentration. And active transport uses energy, whereas diffusion does not. Active transport also uses carrier proteins in membranes, which diffusion does not. 2. Explain the importance of microvilli shown in figure 1.2. So microvilli will increase the surface area, which will increase the rate at which nutrients are being absorbed from the intestines. C. Goblet cells provide protection for the epithelial cells that line the intestine. 1. State the name of the protective substance produced by the goblet cells, that is mucus. 2. Suggest why a protective substance is necessary in the intestines. So mucus, the protective substance in this case, will prevent the enzymes in the small intestine from digesting it, and it also provides protection for the intestine against pathogens. Describe the roles of lacteoles. So lacteoles are responsible for absorbing fatty acids and glycerols that have been produced following the digestion of fats or lipids, and these lacteoles will then transport them to lymphatic system, because lymphatic system is the transport is how the lipids or fatty acids and glycerols are transported throughout the body, as they cannot be transported in the bloodstream like other nutrients. E. Complete table 1.1 by identifying the level of organization of each structure. Choose your answers from the list. Each word or phrase may be used once, more than once, or not at all. Gallbladder is an organ. Endoplasmic reticulum is a cell structure since it's found inside of a cell. Intestinal epithelium is a tissue since it is a group of similar cells working together. And ileum is an organ since it is part of the small intestine. F. Many fungi are decomposers that feed on dead plants. The fungi secrete enzymes to digest large molecules. Students made an extract from a species of fungus. The extract contained digestive enzymes. The students carried out an investigation to find out if amylase and pectinase were present in the fungal extract. They made agar plates by filling petri dishes with agar jelly containing either starch or pectin. So starch would be digested by amylase and pectin would be digested by pectinase. They cut four holes of the same size in the agar jelly in each petri dish. The holes in each petri dish contain the same volume of 1% amylase and 1% pectinase solution, as well as distilled water and fungal extract. The petri dishes were kept at 27 degrees Celsius for 4 days. After this time, a dye was poured into each dish to stain the areas where starch and pectin remained. So the area where starch and pectin remain would be the area that they have not been digested, and the other area where they did not remain would be the area where they were digested by the enzymes. Figure 1.3 shows drawings of the stained agar in petri dishes. The clear zones indicate the area where no starch or pectin remained. So in the petri dish, which contains starch and agar jelly, the enzyme amylase will digest the starch and create this clear zone, which is the area which does not have starch. So as you can see, the areas a clear zone is observed around solution A and D, which means that A and the holes A and D have the enzyme amylase, and A was 1% amylase, whereas D was produced using the extract. As the clear zone around D is greater, this means that the amount of amylase enzyme in the fungal extract is greater than the amount of enzyme in A. So the amount of enzyme in fungal extract is greater than 1%, which is why it produces a larger area by digesting more of the starch. In this petri dish, which contains the pectin in agar jelly, we can see that the clear zone grows around solution B and D. So D again has fungal extract, which contains pectinase enzyme, and B was 1% pectinase enzyme. As you can see, the pectinase enzyme will digest pectin and create the clear zone. This clear zone is smaller around D compared to whole B, which means that the 1% solution concentration is higher compared to the pectinase enzyme concentration in the fungal extract. One, state what conclusions can be made about the enzymes in the fungal extract and give evidence from figure 1.3 to support your conclusions. Fungal extract contains amylase and pectinase, both of the enzymes, as there's a clear zone around the whole D in both of the petri dishes containing starch and pectin. Concentration of amylase in fungal extract is greater than 1% due to the larger clear zone in this dish, and concentration of pectin in fungal extract is less than 1% due to a smaller clear zone compared to the 1% pectinase solution. Fungal extract has higher concentration of amylase than pectinase as a result, and this is indicated by a larger clear zone around the fungal extract in the petri dish which contains starch. 2. The investigation was repeated at 5 degrees Celsius instead of 27 degrees Celsius. Predict the effect of the lower temperature on the results. So at lower temperature, enzymes will work slower, and that means less of the starch and pectin will be digested, which means the clear zones will have a smaller area, and the pattern of the clear zones will remain the same. 2a, a scientist investigated sexual reproduction in flowering plants. Figure 2.1 shows the procedure for crossing two plants of the same species. So anthers are removed and paintbrush is used to pick up pollen from another plant. Stigma is ready to receive the pollen, and the pollen from the second plant are transferred to the stigma of this plant, which is then covered by a bag. The seeds will develop as fertilization occurs. The scientist collected the seeds and germinated them. The leaves and the flowers of the offspring plants showed phenotypic variation as they were not all identical to parent plants. The scientists then investigated the chromosomes of all offspring plants and found that they had exactly the same number of chromosomes as the parent plants. 1. Defined the term chromosome. So chromosome is a molecule of DNA which contains the genes. 2. Suggest why the scientists placed a bag around flower 1. This will prevent pollination as it prevents pollens from another plant landing on the stigma of the covered plant. 3. Explain how sexual reproduction results in the variation that the scientists discovered in the offspring plants. Sexual reproduction involves meiosis, which produces gametes that are genetically different as they'll have different alleles of the same gene. So they can have the dominant allele or the recessive allele of the same gene. As an example here, if you've got two heterozygous parents that have a dominant and a recessive alleles, they'll form these four gametes, each one of them having a single allele of the same gene, and these alleles can randomly fuse together, which you can use the Punnett square to predict the genotypes, and you'll get these three different genotypes. And having these different genotypes means that there is genetic variation. So meiosis will produce genetically different gametes that will fuse together randomly, and this will lead to genetic variation. For the chromosome number of the offspring plants is the same as the chromosome number of the parent plants in this investigation. Explain how the chromosome number is maintained from one generation to the next. So gametes are produced by meiosis, which will produce haploid cells. So they have half the number of chromosomes, and when these gametes fuse together at fertilization, a diploid cell will be produced. So it will again have the complete set of chromosomes. B. The plant Camellia japonica has flowers that can be white, red, or a mixture of these two colors. When red flowered plants are crossed with white flowered plants, all the offsprings have flowers with petals that are a mixture of red and white, as shown in figure 2.2. So when an offspring plant has the phenotype of both of the parents, or an individual with a heterozygous genotype expresses phenotype of both of the genes, this is called codominance. And you can also tell that by seeing how these symbols are written with a subscript for each of the different alleles. The gene for petal color is given the symbol P. The allele for white petals is given the symbol PW, whereas the allele for red petals is given the symbol P with subscript R. Table 2.1 shows the phenotypes of 3 different pairs of parent plants. Complete table 2.1 by giving all possible genotypes of the offspring plants that could be produced by these parent plants. And you're given the space for working over here. So in the first case, you've got red petal plants. In the second one, you've got white and red. In the second one, you have got parents that are heterozygous. So they have the allele for both red and white petals. In the first row, both parents have red petals. So both of them need to have 2 alleles for the red flowered petals. So here I've drawn the Punnett square. You can draw all of this in your space for working. So in the first case, all of the offsprings will have red petals, since all of them will have 2 alleles for the red petals. In the second row, you've got one parent that has red petals. So they've got so this parent has 2 alleles for the red petal, and the second parent has white petals. So they'll have both of the alleles for white petals. In this Punnett square, you can see that all of the offsprings will be heterozygous. In the last row, you've got both parents that have red and white petals, and both of them will have 2 different alleles, one for red, one for white, and their offsprings will have the genotypes as shown in this Punnett square. And here I've written down all of the genotypes. Just make sure that you write the subscripts in the format that they are shown as in the question. I wasn't able to do that, so I wrote them as it is. And as for the inheritance, that will be codominance, since both of the alleles are expressed in the phenotype of a heterozygous individual when both of them are present. Figure 3.1 shows changes in the concentrations of hormones FSH and LH during a menstrual cycle. A1 suggests the target organ of FSH, that is the ovary. 2. State how FSH reaches its target organ, so it's dissolved in blood. 3. Describe the relationship between the 2 hormones in figure 3.1. So as you can see, the level of the concentration of both of the hormones will rise and then fall. The peak value of LH, indicated by the cross, is higher than the peak value of FSH, indicated by the small circle, and LH has a lower concentration during the initial stage or before the peak, and it also has a higher concentration after the peak value. You can mention that with using the data as well for 2 marks. So I talk about how both of the concentrations increase and then decrease. Both of them have peak concentrations at day 14. B. Describe the roles of FSH and LH in the menstrual cycle. So both of them are secreted from the pituitary gland. FSH will stimulate the development of follicles in the ovary and the development of egg cells in the ovary. This is during the initial stages of the menstrual cycle. FSH will also stimulate the secretion of estrogen from the ovary. Again, this is before day 14. Now, close to day 14 or at day 14, LH will stimulate ovulation, which causes the egg to be released from the ovary when LH concentration peaks at around day 14. Here we've got this. And then LH will stimulate the corpus luteum, which is the empty follicle. Corpus luteum is the empty follicle which forms after ovulation when egg has been released from the follicle. So LH will stimulate corpus luteum to secrete progesterone, and those are the roles of the two hormones. Changes that occur in the lining of the uterus during one menstrual cycle. So during the first stage, the thinning of the lining occurs between days 1 to 7, which is the stage where the endometrium sheds off. And during the second stage, the lining of the uterus will thicken up before ovulation. So after 7 days, the lining thickens as FSH is being secreted, and FSH will stimulate the secretion of estrogen. And it's this estrogen which causes the lining of the uterus to thicken up. And this happens up until ovulation. After ovulation, the lining stays at the same thickness, and this happens after day 14. So progesterone is being secreted, which maintains the level of thickness. Now, if the egg which has been secreted, the egg which has been released at ovulation, if that egg is not fertilized, the corpus luteum will disappear and progesterone will not be secreted. So you'll get back to the first stage where the lining of the uterus will thin out during the first 7 days of the menstrual cycle. The oral contraceptives are a method of birth control taken by women. Outline how the hormones in contraceptives act as a method of birth control. These contraceptives have progesterone, which will decrease the secretion of FSH and LH. And as we saw earlier, FSH and LH are responsible for the development of the egg cell and the follicle, and also the secretion of egg at ovulation. So if progesterone is going to decrease the level of FSH and LH, this means that ovulation and development of eggs will not occur. When eggs will not be released, then they will not be able to fuse with sperm cells. Four involuntary actions occur because nerve impulses travel along the components of reflex arcs. An example of an involuntary action is the rapid movement of a hand after unexpectedly touching a very hot object. Figure 4.1 shows the structures. Figure 4.1 shows the structures that are involved in the movement of the hand. And in part A, you've been given table 4.1, which shows the functions of some of the structures. In figure 4.1, the names of the structures and the letter from figure 4.1 that identifies each structure. You are supposed to complete this table. So the function of the first structure is to conduct impulses to the central nervous system, that is the sensory neuron. And the structure which conducts impulses to an effector is a motor neuron. The structure which conducts impulses only within the central nervous system is a relay neuron. And the receptor's function is to detect stimulus. Alright, so U over here is the receptor, indicates to the receptors in the skin cells. And V, as you can see, is carrying an impulse away from the receptor and towards the central nervous system. So V has to be the sensory neuron. Z over here is the effector, since this is the muscle that carries out the response. Y over here is the neuron which is carrying impulses away from the central nervous system. So Y has to be the motor neuron. And X is the neuron inside of the central nervous system. So this is the relay neuron. Here I've got the letters of the structures, and Z, as we said earlier, is the effector or the muscle which will carry out the response. B. Figure 4.2 shows the structure of the synapse at W. On Figure 4.1, describe how an impulse travels from the synapse shown in Figure 4.2. So let me just drop out a few terminologies here. Here, this is the postsynaptic neuron, and you'd know that, as you can see, these vesicles containing neurotransmitters in this neuron, and this is the postsynaptic neuron because it has receptors. The synapse will include the membrane of the presynaptic neuron, which is also called as the presynaptic membrane. The synapse will also include the gap between the two neurons, and it will also include the postsynaptic membrane. This combination is known as the synapse, and the gap alone between the two neurons is called the synaptic cleft. And the way that the impulses travel across the synapse is that the vesicles in the presynaptic membrane in the presynaptic neuron will diffuse with the presynaptic membrane, and this will release the neurotransmitters into the synaptic cleft, which is the gap or the space between the two neurons. Then neurotransmitters will diffuse across the synaptic cleft, and they'll bind onto these receptors on the postsynaptic neuron, and this will generate an electrical impulse in the postsynaptic neuron, which will travel on. C. State one example of a reflex action that occurs in the eye. I've written down blinking as an example. Five A. State two factors that affect the volume of urine produced in the human body. This could be dependent on the temperature of the environment as well as the volume of liquid intake. B. Nitrogen is an important element for organisms. In a livestock farm, waste from animals contains protein. This waste is often spread on farmland as a fertilizer. Describe how the nitrogen in protein is recycled in the soil into a form that plants can absorb and use. So plants can only absorb nitrogen from soil where it's present in the form of ions, because plants cannot absorb the unreactive nitrogen gas which is present in the air. So you're supposed to describe how the protein in the waste of animals is converted to nitrate ions. And this happens when protein in the waste is broken down to amino acids in the soil, and amino acids are then converted to ammonium ions by decomposers like fungi or bacteria. The ammonium ions are then converted to nitrate ions by nitrifying bacteria, and the plants will then be able to absorb nitrate ions as well as ammonium ions. Sea waste from livestock farms often pollutes water courses such as streams and rivers, leading to a reduction in biodiversity. Explain how the pollution of water courses by animal waste leads to a reduction in biodiversity. The waste of animals will cause eutrophication because the waste of animals will be used as fertilizers, and these fertilizers in the soil will cause leaching out of nutrients like nitrates when it rains, and so the water will leach out the ions into the water courses, and this causes an increased growth of algae and green plants, so much so that these will cover the top of the water, and this prevents sunlight reaching the plants that are present underneath and cannot reach the surface, and those plants are going to die. Decomposers like bacteria are going to increase as a result and use up any remaining oxygen which is dissolved in the water. As this will decrease the concentration of oxygen in the water, other living organisms will die because they will not be able to get adequate amount of oxygen for aerobic respiration. As different kinds of organisms are lost by their death, the biodiversity will reduce. 6. Enzymes are catalysts. A. Define the term catalyst. So a catalyst is a substance which increases the rate of a chemical reaction and is not changed by the reaction. B. Figure 6.1 shows diagrams of 3 enzymes and 8 different substrates. 1. State the letter of the substrate that will be broken down by enzyme 1. So enzyme 1 over here has an active site which is curved almost like a semicircle. So this means that the substrate for enzyme 1 should have a similar structure. which happens to be T. To explain in terms of enzyme structure the reason for your choice in 6b part 1, so T has to be able to bind to the active site on enzyme, which means the active site of enzyme has a complementary shape to T. C table 6.1 lists some enzymes and the reactions that they catalyze. Complete table 6.1. So maltase enzyme catalyzes the breakdown of maltose to glucose, and the enzyme which catalyzes breakdown of proteins to amino acid is protease. You can name specific protease enzymes as well. Lipase will break down fats to fatty acids and glycerol. And the breakdown of lactose to simple sugars is catalyzed by lactase enzyme. The insertion of a short length of DNA into a plasmid is carried out by DNA ligase. And restriction enzyme will cut the DNA strands. And this is it. We are done with this paper. Thank you for watching.